Assalamu alaikum. I hope so you are doing fine. Let us discuss the pictures or the slides that you will encounter in your practical examination for the histology first year BDS. So these are the slides that I will be discussing with you today. Uh, cell, well, starting from epithelium, glands, connective tissue, all the way down to the respiratory system. So let us go through them one by one. So starting with the epithelium. Okay. So, quick go. Epithelium is surface epithelium is simple or stratified. Simple has one layer, stratified has multiple layer, and in the simple one there is squamous, totally flattened, cuboidal, cube shape, both sides equal, columnar, one side longer than the other, and pseudo-stratified. It means that it is actually single layer, but the cells are of different sizes, so they don't seem of the same level. So, in the cuboidal, you cannot see usually the cell membranes, but nucleus will help out. In the squamous, you will see these flat nuclei. In the cuboidal, you will see these rounded nuclei. In the columnar, you will usually see these oval nuclei. And in the pseudostratified, you will see nuclei at different levels, like shown here. So let us go through the pictures one by one. Okay. So here is the picture of simple squamous. Simple squamous is a very tricky picture for the because it is in a single picture it is very difficult to get out. So here are these are the simple squamous cells. The one that is being shown right here, these one. So these are the flat nucleus. This is the single cell. This is another cell, and so on. So this whole line is the simple squamous epithelium. So sometimes these pictures are shown, and other times you are shown pictures like this where the uh, epithelium has been spread out like a paper, and a picture from a top is given. So this is a picture that you are sometimes shown, and here is one of the slide up one of the poorly produced slide. But as you can see, this fold has folded upon itself, but it is a simple squamous cell. So these are the cells actually. This is a cell that has folded upon itself, and so on. Okay, let's move on. Then comes the simple cuboidal. These mostly are very self-explanatory. Cuboid, simple squamous, sometimes you are given this and sometimes you are given other pictures. Okay, simple cuboidal. You may not see the individual cell shape, but you will see the oval of the nucleus, like this rounded nucleus that you will see. So this rounded nucleus, this gives an idea of the simple cuboidal epithelium. Single layer in cuboidal in shape. So since this is simple cuboidal epithelium, okay. And here is the picture that is showing it right here. Simple cuboidal epithelium. You may not see very clearly. This is a simple cuboidal epithelium. So these are actually the cells. This whole slide is not, but these are actually the cells that are the cuboidal ones. This is one cell and this is the other cell. So that is why I'm showing you the other pictures too. Okay, moving on, simple columnar epithelium. Simple columnar epithelium, let me show you here again before going into that. Yeah, so this is a columnar cell. This is one columnar cell. As you can see, this is one cell. This is one cell. These are one cell. This is one cell with its nucleus right here. So this is what you will see here. So this is another columnar cell with its nucleus, and you can see all of the nuclei are in the same line. So that is the thing that makes them separate. And in this picture, so you will see the same thing here, and here due to zoom restriction you may not be able to identify it, but here you can very clearly see the nucleus in the same line. That's a very clear thing that is visible, and then this is one cell, and this is one cell, and so on. Okay, moving on. Simple cuboidal we have seen, simple columnar we have seen, and then pseudo-stratified epithelium. This is very clear picture that is shown. So here this is a picture. As you can see, these cells are at different levels. So there is one cell right here, and then there is another cell here, then there is another cell right here, there is a cell here, and so on. So these are the cell, and that is due to their different shapes. Their nuclei are at different points. So here is one nucleus, other nucleus, here there another. So these are pseudo stratified epithelium. All of the cells are ultimately reaching the basement membrane, the base in the bottom, and then there are other cells that is not part of pseudo stratified. These are goblet cell. We will see them shortly when we go to the respiratory epithelium. So this is pseudo stratified epithelium, usually found in respiratory tract and in male reproductive tract. Stratified squamous keratinized epithelium and non-keratinized epithelium. Now comes the stratified layers, and in the stratified layers, the key point is that they are multi-layered. And as you must keep in mind that their name is on the base of the topmost layer, not the bottommost layer. So that is stratified squamous, keratinized, non-keratinized. I will show you here. Then I will move on to the pictures in your other folder. This is stratified squamous epithelium. You can see the topmost layer, the cells are very flat. So these are the topmost cells. These are the flat cells. These are the flat cells. So these are the stratified squamous cells. And you can see them here, and you can see them in the next pictures. This is stratified squamous epithelium. This is a squamous keratinized epithelium. Now, what is the difference? In the stratified squamous non-keratinized, the nucleus all the way till top, the cells all the way till top have the cell nucleus. You can see a nucleus here, you can see a nucleus here, you can see a nucleus here, and so on. You can see the nucleus all the way till the top. However, in the stratified squamous keratinized epithelium, there comes a time where the nucleus top and the topmost layer are non-cellular, so non-alive, or you can say that, or they are not having any nucleus in them. So here you can see this is the layer, this is the basement membrane, this is the layer, these have the nuclei, this is the last layer that have the nucleus, and all the way till here you can see there is no cell with having any nucleus in it. So that is the keratin layer, or that is the non-alive layer that is seen here. So let me show you in your pictures. So this is the stratified squamous keratinized epithelium, because nucleus is all the way till here, and in all this layer that is the keratin layer, and non-nucleated layer you can see no nucleus. And then in the next part, this stratified squamous non-keratinized epithelium, nucleus are present all the way till top. Okay, so here is a nucleus. They are present, this is nucleus that is present here and here and so on. So this is epithelium. Now comes transitional epithelium. Transitional epithelium is present in the bladder, and it is usually confused with stratified squamous epithelium. The only difference comes on the topmost layer. The topmost layer, the cells, rather than being flat, they are very prominently dome shaped or prominent. So this is the dome shaped cell. This is a dome shaped cell. These are the dome shaped cell. It is much like stratified squamous epithelium, but on the topmost layer, they are not uh, flattened. You can see here the topmost layers are larger than that. You can see it here also. You can see topmost layer are relatively bigger one as compared to though, and usually they're dividing, and you can see also the binucleated cells you can see here too. So this is the picture, and here the topmost layer dome shaped cells very prominently are visible in this part. This part they are very prominently present. Moving on quickly to the glands. And now these are the epithelium, transition epitheliums. Now there are three types of glands: serous, mucus, and seromucus. Let me very quickly show you the picture so that I can discuss it with you easily. The mucus glands are relatively empty, and they will not shown to be stained here, and their will nucleus will be pushed towards the side with flattened cells. And the serous gland will be colored usually with depending on the gland. It can be eosinophilic or hematoxylin or with eosin, and the nucleus is usually rounded and it's present in the cell. And the seromucous gland is a compromise between the two, is a mixture between the two gland, where inside there is mucus cells and periphery there are serous cells. So here is mucus gland majorly, and in the mucus gland you can see very clearly that these are one acinus, when mucus acinus. So this is mucus gland, and here is the empty cell, somewhere here is the lumen, and these are the flattened nuclei that are present near the periphery. So these are the mucus gland. You can see it here, you can see it here, even if it's not that much flattened, but you can see the individual cells right here, and you can see these nuclei that are visible on the periphery on the each side. But as opposed to this, compare it with the serous gland, you can compare it with the serous gland right here. And I know I have picked very clear pictures. You may not be getting that much clear picture, but you will be able to see the difference. And here you can find out this is serous acinus, and these are the cells that are present, and these are very well stained as compared to that, and nucleus that are present. And last but not the least, the mixed gland. So here is a mixed gland, this is a mixed gland. So this has a mucus gland in the center, and in the periphery it has serous demilume. So there is a mucus gland in the center, and in the periphery there is serous demilumes. So they will have, this one will have a feature of mucus, this will have a feature of the serous gland. So these are the one. So you can see in this part you can only see the serous gland, in this part you can see the serous and mucus gland, and so on. So depending upon the amount, you can see this one is the predominantly serous mucus gland, this one is the predominantly serous gland, this one can be counted as seromucous gland, so this can be counted as seromucous gland. So these are the things that you will be able to see. So this is predominantly serous gland, and here is the serous this is predominantly serous gland. So you can see this is the demilume and with serous cells and so on. Don't be confused by this. These are the white cells or adipose tissues, and they may confuse you to think of it like a mucus cell, but this is simply adipose tissues. See, so don't get confused by this. Moving on, this is it is majorly serous gland. Majorly, you can see serous gland, that will be a right answer. And here, this is the mucus gland. This one is the mucus gland. Uh, this one is the mucus gland. So on the basis of that you can see it mixed gland, this is the mucus gland, this is the mixed gland. On the basis of this you can say it mixed gland with, but this is more serous component in it, so that is the thing to be focused here. Mammary gland, this is a gland of the breast tissue. You will see stroma, the connective tissue that is present between, this is the stroma, and these are the acini. Depending upon the, okay, you will see these lobules you will be seeing, and in that secretory component will you will here that will be usually cuboidal and sometime male component that is present inside them. So these are the things that will help you figure out uh, the mammary gland. Okay, now moving on. Loose connective tissue, as the same name indicates, it is the connective tissue that is loose, and it has a very less number of cells and fibers, and majorly it is composed of ground substance with fibers between them. So it is a very characteristic appearance. Usually cell nuclei that are placed very far apart from each other, and there is fiber running between them, so it is very easy to identify. So that is the loose areolar connective tissue. See, so this is the loose areolar connective tissue, and then now this brings us to the adipose connective tissue. White adipose tissue. White adipose tissue has very characteristic soap bubble like appearance, or individual cell will have signet ring like appearance, where each of the cell is basically an empty cavity. So this is one cell, this is another cell, this is another cell, this is another cell. So things like that, I will help you figure it out. So this is white adipose tissue. You may see something like this too. Sometimes when it is very light, so it seems like this. So this is one cell, and it is signet shape appearance because this is a ring with that is its nucleus, that is its nucleus, or um, diamond in the signet ring. So that is that adipose tissue. Brown adipose tissue, it has very small loculi, but I don't have very good picture for it right now. So I can show you, but I don't think you have that. Like, yes, that's a good picture. So here you can see each of the cell has very large number of small loculi rather than one cell having one large common fat droplet. So this is an example of white adipose cell where is a large droplet, but this is an example of the other is a brown cell. So these are very small, very large number of brown droplets that are being shown here. Okay, dense regular connective tissue. These the collagen fibers are arranged parallel in bundles. It is very easy to show you the bundles, and between the bundles you will see the nuclei that are present between the bundles, and the each of the bundle will not have any nucleus, but between the bundles you can see that this nucleus that will be present between the bundles, and this is a particular site that gets confused with the skeletal muscle fiber. Be aware of it. I will show you them when we reach there, but they have striations, and they are basically larger bundles. That is dense irregular connective tissue. Very easy to identify picture dense irregular connective tissue because they are cut at different angles. So you will be seeing like this one that is being cut at in a long direction, and the other one that are cut in the horizontal direction, the vertical direction, like this one or this one. And so these are the pictures that you will see in there. Now blood. Blood, I don't think so in the exam you will be asked to identify any particular cell. So blood will smear will consist of a large number of red nucleus with uh, I'm not going into the detail of the white cells that are present between them. So all of these cells are red blood cells, and due to their biconcave nature, their periphery is darkly stained as compared to the center. And white blood cells, the different types that I'm not going to discuss here, can be seen in different type places. Multipolar neuron. This is regarding the nerve. Mm, Let me show you here. The nerves are of different types, pseudo-unipolar, multipolar. They're usually the one that is easy to give in the examination is this one, the multipolar neuron in the spinal cord. So this is actually a picture from spinal cord. Let me try to have a good picture of spinal cord right here. So this is a picture of the spinal cord, and from there you had gotten the multipolar neuron. This is a spinal cord with the H shape, gray matter and white matter in the periphery. And here you will see the large nuclei and the anterior horn cell in this part, the anterior part. In the anterior part you will see, in this part you will see these cells. These are the multipolar neuron. So these can be labeled as the anterior horn cells two in this particular picture. um, Let me show you as we go to the higher magnifications. So in the higher magnification, you usually see this picture like this. So here is the alpha motor neuron. These are the cells that are present. So this one, so this is the one that is shown you right here. This in the, and to identify and differentiate it from the other types of cell, you can sense the granular appearance of this granular appearance is the Nissel's substance that help you identify the nerve related cells. Nerves, okay, let me get here peripheral and nerve in the cut in different directions. This is the brain, spinal cord, ear. I think I have peripheral nervous system in the end right here. Yes. Okay, so let me show you first the cross section and then the longitudinal section. In the cross section, the peripheral nerve, it consists of axon in the center with its myelin sheath in the periphery. So you will see a large number like this, and you will see this axon in the center and its myelin sheath in the periphery. Axon in the center and myelin sheath in the periphery. And this picture, it's not a very good example, but in most of the pictures you will see the axon and the myelin sheath in the same color. Let me show you here, and that is how you will differentiate that it is not actually the nucleus, it is the axon, because it is being stained by eosinophilic color too. So this is the axon, that is the Schwann cell, and the area myelin, that is basically lipid, so that is being washed out. This myelin is washed out, so you will see empty spaces. And this is one nerve axon, and this is an axon and covered by its myelin. So this is endoneurium technically speaking, and then there is a whole bundle. As you move out, you can see this is endoneurium. So this is the whole bundle, and this is the whole bundle. And let me do it again. So this is the whole bundle, this one, and the line that is covering is the perineurium. And as you move out, you can see here, as you move further out, you will see the covering of the whole. This is the perineurium. So around each is endoneurium, this is the perineurium, and around that is the epineurium. So this is technically the epineurium, this one. Okay. This is the nerve, and when we see the longitudinal section, this becomes a little tricky when you focus it, and you will have to look at it in a little depth. You maybe identify axons, and they're surrounded by myelin sheath. So this is a picture that gets confused with the dense regular connective tissue. um, I may not be able to show you very clearly here, but like this is the axon. Here you will see like these these nodes of Renvier you may be able to identify. Mm, I don't have some picture of my, but that's good enough pictures. So here is the actual, here's the thing, that you will see the axon. This is the axon running in the center, this one. This is the axon running in the center. Let me go to do that again. This is the axon running in the center, and this is the endoneurium in the periphery, and there is a space that is white in between them, that is the lipid layer between them. So here you can see it again, the axon running in the center, and this is the lipid layer, that is the endoneurium that is around them. So in between them it is very clear, this is the axon running in the center, and this is the endoneurium around it. And these nucleus that you are seeing, that are not related to the nerve cell itself, but these are actually the cell bodies of the nuclei of the Schwann cells, or the covering, that is the endoneurium, and that is basically the perineurium, the bundle that is covering the nerve bundle. So that is about the nerve. And let me move on. That is the longitudinal section, and you will see this is the nerve bundle, and that is the perineurium in the entry. And so this is a very good picture of cross section. And don't get confused by these maybe, because these are the fibers that are not cut in the exact perpendicular position. Autonomic ganglion and dorsal root ganglion. So this is a dorsal root ganglion. If you get lucky enough to get a slide that is zoomed out, that is usually attached to one side. But a very good thing that is helping you is that in dorsal root ganglion the cells are present in clusters. Like these are the cells that are present in a group, these are the cells present in a group, here, and the nerve bundles that are running as bundles between these clusters. The nerve fibers are running between these clusters. There is one thing, and if you zoom on, and you will be able to see that this here, the cell nuclei are usually present in the center. So cell nuclei are present in the center, and you can see in most of the cases the cell nuclei are present in the center, and they are surrounded by a very regular layer of cells. So their surrounding cells are very regularly spaced, or they're at least more than that which you will see in the autonomic ganglion. So this is the dorsal root ganglion. And if you move on to see autonomic ganglion, this is the dorsal root or spinal ganglion. And if you move on to see the autonomic ganglion, this sympathetic autonomic ganglion consists usually of sympathetic ganglion, because parasympathetic ganglion are in head and neck, in the abdomen they are not present. You can see there is not that much grouping between them, and these cells are widespread apart with large number of bundles passing through them. Here you can see, and one more thing, that a large number of nuclei, these are pushed to one side. The nuclei are pushed towards one side. So these are not present in bundles, and fibers are passing through each of them. And you can see right here, you can see right here, you can see right here, a large number of cells will have nuclei that are pushed towards the periphery. So this is regarding the dorsal root ganglion, and so this becomes the dorsal root ganglion, autonomic ganglion, sorry. And you can see the cells are widespread apart, and this is again the autonomic ganglion, the cells are widespread apart, and this is the dorsal root ganglion. You can see the clusters of cells that are present in them, and the bundles that are going between them. Then there is a spinal cord. I think I have shown you spinal cord, but I will show them again. The spinal cord, you will see it in different zoom levels. If you are lucky enough, in your slide you will see a picture like this. It's a very typical picture of spinal cord, showing the butterfly shape of the gray matter and white matter outside. But sometimes you may get a picture that is a little more zoomed into the anterior horn, and that is zoomed into the central canal. So that is what has happened in this particular picture too. So you have been given a picture that is focused on the center, with the central canal surrounded by cuboidal cells, and there is the gray commissure, that is the spinal cord. Thymus, and these are endocrine glands. So let me first walk you through these cells, different organs. These are very similar to one another in some aspect, but different. First of all, be very vigilant about the difference between lymphoid nodules, because all of the autonomic ganglion will have these lymphoid nodules. So I will walk you through them one by one, and then we will go on. So easy one, I think, for to start is lymph node is a good place to start. Okay, a lymph node has lymph nodule capsule in the center, lymph nodules in the periphery, and capsule in the outermost layer, and medulla in the center. So you will have a picture somewhat like this. You will see capsule that is in the periphery, and then you will see capsule in the periphery, and you will see these lymphoid nodules that are present in the below the capsule. And this, the solid areas, these are medullary cords, and these empty areas, these are sinuses. So medullary cords and sinuses you will be seeing there. And very good telltale is this space between the capsule and the peripheral nodules. So this is the capsule, this is the pericapsular space, this is the cortex with nodules, this area is paracortex, and in the center there is medulla. So these are the things that you will be seeing in the lymph nodes model. Lymph node is a whole organ, it contains lymphoid nodules. And here I have seen students make that mistake again and again, that they consider lymph nodules and lymph node one and the same thing, that leads them to misidentify the tissues. So these are lymph node, and you will see different pictures of lymph node. This is at the zoomed out level. You will see this is majorly focused on medulla, this is medulla and cortex, and this is the capsule and the medulla cortex with nodules and pericapsular space, and this is the medulla with cords, and this is another picture of zoomed in picture. Other slides that we have, you can go through them in your free time, which you don't have much. Then there is a spleen. In this you will see one thing, you will see these areas, some areas, dark areas where cells are concentrated, like this one, like this one, like this one, and the other will be the rest of the areas. Like all the circle areas, these consist of the lymphoid nodules where cells are concentrated. And in the other one is, this is called, this one, the all of the circled areas is called white pulp. This is the white pulp, the all of the rest is the red pulp. So a white pulp and red pulp. And one very interesting thing that can help you identify spleen immediately is if you can look at the nodules. In most of the cases you will be able to identify an artery running inside the lymphoid nodule. So this is the lymphoid nodule, this is the central artery. That will help you identify the lymph node very easily, spleen very easily. In most of the lymphoid nodules, you can see here is the central artery. I know it's not in the center, but it is there. So these are the ones that will help you. This is the central artery. This is the central artery, and that will help you identify the particular. This is the central artery, like here, this is the central artery. So they usually are not present in the center, but they help you identify the spleen very easily. White pulp, red pulp with central artery. Let me show you the picture. This is one lymphoid nodule with a central artery that is present right here. And moving on, there is a white pulp, and there is the medullary cords and sinuses that are present here too, not going into the detail. So that is medullary cords and sinuses. Then there is thymus. Thymus, one very important thing, it does not have it has different lobes. So this is one lobe, this is the next one, this is the next one, this is the next one, this is the next one. So it is divided into lobes and different segments, and in each of the segment there is an outer area of this. In each segment there is an outer area, this one, this is the outer area of cortex, and inside there is medulla. So there is outside cortex, inner medulla, outside cortex and inner medulla. Don't get it confused, sorry, this is cortex and this is medulla. So don't get it confused with any other. This is a very easy one that you can help to, that you can find out. So, and inside the medulla you will see these things in lower magnification, and I'll show you them in higher magnification too. So these are Hessel's corpuscle. This is cortex, this is cortex, this is cortex, and this is medulla. And in the medulla you will see these things. These are the Hessel's corpuscles that helps you identify them. So this is thymus, and last but not the least, that is palatine tonsil. That is the part of mucosa associated lymphoid tissue malt, and that contains lymph nodes, lymphoid nodules. So they may get confused with the lymph nodes too, but unlike lymph node, there is no capsule. You can see the outer layer, but if you focus a little, if you concentrate a little, you can see the outer layer is consisting of stratified squamous keratinized, non-keratinized epithelium. So it is epithelium. So this is a stratified squamous non-keratinized epithelium that is covering in the back of the pharynx. So that is the part that will help you differentiate between this and Lymph node. It is not lymph node because it does not have medullary area. It is not lymph node because it does not have capsule of dense irregular connective tissue, but it has certified squamous non-keratinized epithelium on it, and as opposed, it does not spleen because the nodules have central artery in them, and thymus was very self-evident. So let me go to the different pictures. So this is higher up, you can see, and yes, these are called the crypts. These dips, these are called, this is the crypts. These are the folds that give it more surface area. So there is a steadfast squamous non-keratinized epithelium that I was talking about in a little zoom image. So this is the nodule, this is the zoomed out picture, and in the opposite end you may see a capsule, but again, if you get even a slide that is focusing on its back aspect, from this is the point where it was dissected out, and this is the capsule part, that is the, you can see that is not epithelium, but you don't see any uh, subcapsular space as you saw in the lymph node. So this is about that, a lymph nodule and palatine tonsil. So let me quickly go through them. This is the thymus, one, and this is the Hessel's corpuscle in them. As you move down, this is the zoomed in thymus again, that is the, here is the Hessel's corpuscle in them. As you further move down, there is lymph node, and lymph node, these are the lymph nodule that are present inside them, and this is the pericapsular space. This is the empty area that is surrounding between this and this. Outside this is the capsule area. So that is it. Lymph node, that is the spleen. It has this is the white area, and this is the white pulp, that is the lymphoid nodule that is present, and in between them you will see this central artery that is present here. So that is the spleen, that is the palatine tonsil. Here is a little torn up, but you may be able to appreciate some. This is the crypt that helps to identify, and the covering epithelium that is here. If you get zoomed in, or you can see more, you can see, yes, you can see here, this is the epithelium. Not very clear slide, but still you can see the flattened cells on the top, and this is the epithelium that is covering it. Come to the muscles. Skeletal muscle are arranged in straight bundles, and if you zoom in to them, and they have striations not very clearly visible in this particular picture, but they are there, and their diameter is much more than the dense regular connective tissue. And one more thing is that in the bundles in the periphery there are nuclei. The nuclei are not present in the center. That will help you differentiate it from the uh, cardiac muscle. So this is the cross section, this is the cross section, this is the cross section. In each of the picture you can see the nucleus are present in the periphery. All in all of these pictures the nuclei are present in the periphery, they are not in the center. And here is a longitudinal section, and here is the nucleus present, here is the nucleus present. So all of the nuclei that are present in the periphery, not in the center. So here you can see these nuclei are present in the periphery, not in the center. So these are the things that will help you identify skeletal muscle. Ideally speaking, you should see striations, but that are visible in a very clearly made tissue sections, in a very nicely made tissue slides, but they are not always the case here, not in this picture at least. So skeletal muscle again, fibers are separated. Again you can see the the nucleus, you can appreciate the nucleus running along the peripheral wall. Cardiac muscle. Okay, let me move on to another slide of cardiac muscle. Cardiac muscle, you will see branching and in them, and muscles making connections with one another, and if you're lucky enough you will see intercalated disc. So this is how. And one more important thing, the skeletal muscles, they have nuclei between them. So here is a muscle. These are the intercalated disc. This. These are the intercalated disc, and then cells are the nucleus are present inside the center of the cell. So this is the cell going on. This is the intercalated disc between them, and if you follow through, this is the nucleus, and nucleus will have a empty area between them. And if you focus a little, you may be able to see branching too, like here the here is the intercalated disc, cell is going on, and here is being branched, so you may see the branching too. So these are the things that will help you identify the cardiac muscle. Let me move on to this again. So here, if you are not even seeing the sessions, you can see the branching as this branch goes along. In the cardiac muscle, in the skeletal muscle, there is no branching. The single muscle will go on. So the branching will help you identify. And the nucleus, here is the nucleus that is present in the center. And you may see, if you focus for long enough, you may see intercalated discs between them too. Like here is intercalated disc present between them, so here is intercalated disc present between them, so you may be able to see the disc. If you cannot see the disc, here is the disc between them, you can see the branching, and that's another way to go. And the nucleus that is present in the center of the cell rather than in the periphery as was in the skeletal muscle. And then there is a smooth muscle. Smooth muscle are typically very small, like these are these like it's like this. So these are the cells, and if you these spindle shaped cells like this, and if you see them in the longitudinal section, you will see a bunch of flattened nuclei irregularly placed in them. So this is the longitudinal section, and if you cut them like this, you may see some cells that are big, some are small, some are big, some are small, some are big, some are medium sized. And in the big one you will see nucleus in the center, and the empty you will see empty in the small one. So this is the longitudinal sections of the, oh sorry, cross sections of the smooth muscles. Let me try to figure it out here. You see, this is a part where the cells are cut in the cross section. So this is a cross section cell between them. This is the tip without nucleus. This is a cross section with nucleus between. This is a cross section with nucleus between them. This is a tip, and so on. So this is the this is the cross section, and this part, this is the longitudinal section. So this is a spindle shape. This is a spindle shaped cell with the nucleus between them. So you will see cigar shape or columnar shape nucleus between them. This is a spindle shaped cell that is going on. So this is a cell that you may be able to see, and nucleus in them between. So these are the cell that is the longitudinal sections of the uh, smooth muscles. I know I'm going a little haphazard. It could have been a little more organized, but um, okay. And then there is cartilage, haline cartilage. Haline cartilage, you will see there is a cell aggregates. So this part is called the perichondrium, and here you will see fibrous tissue, and in the perichondrium you will see flattened cell. In here you will see cell aggregates like this one and this one, isogenous group of cells, and they will be surrounded by a little darkly stained area, that is the territorial matrix, and between them there is very interterritorial matrix, and it is usually a very clean slide, and there is no fibers running between them. So that is haline cartilage. Let me try to show you here too. Yes. So in this particular picture that are a little clearly shown, and these are the isogenous groups. So this is an isogenous group, this is an isogenous, smaller isogenous group, this is an isogenous group. The dark stained area is the territorial matrix, the light stained area between them is interterritorial matrix, and this all area is the perichondrium. Okay, this part area is the perichondrium. So that is haline cartilage. It is usually shown you in the picture of larynx. It's in the trachea, it's very easy to show you this area is haline cartilage, this area between them, this is the haline cartilage. This is the haline cartilage. You can see the very small perichondrium is present here. That is the hyaline cartilage part, and that's perichondrium again. It depends upon the exact slide which you are seeing in subconchus. Um, I think I'll have better pictures. Sometime you may get a tissue slide like this one too, where the perichondrium it's not only the center you will have been focused, and then there is this pictures like this again. The hyaline cartilage, the same thing, but this is a little different from hyaline cartilage. This is elastic cartilage. How did you differentiate? There's only one minor difference between them. You will see these fibers running between them, these fibers running between them. Like, see, if I will remove this for you to focus on. So only the difference that you will see is these fibers that are running between them, like this one, these fibers running between them. So otherwise there is the same group, same territorial matrix, same interterritorial matrix, and so on, but that's a difference that may help you identify. So these are the fibers running between them. So these are all the hyaline cartilage or the elastic cartilage. This was the hyaline cartilage. There is no fibers between them, it's uniform, but this is the elastic cartilage with fibers running between them. This is again an example of hyaline cartilage, that is the articular. This is the spongy bone between below, and the cartilage above, that is making the tip of your long bones for making the joints. So this is the hyaline cartilage. This is the not very well preserved picture of elastic cartilage, where cell have been destroyed but fibers have been preserved. But fibrocartilage cell has been destroyed but fibers have been preserved. It is much like dense regular connective tissue, but there is cells between them. So actually there were supposed to be cells in these spaces. There were supposed to be cells in these spaces, but they have been destroyed due to some issue in tissue processing. But here you will see the fibrocartilage. I think I should show you a picture of fibrocartilage right here. What is this supposed to look like? It is much like dense regular connective tissue, and it has running fibers of these isogenous group are running as a line, not as a bundled up group, but as a straight line between them. So that's usually the difference between them and the other cartilage is again much like that of. I don't have a good picture here, but that's a cartilage. An elastic cartilage, you may see some specially stained pictures where fibers are accentuated, like this one. That is, this is done by special stain. Okay. So here is again elastic cartilage. You can see the prominent fibers that are running between them. So these are the fibers. This is the fibers running between them. So this is elastic cartilage. Now comes the bone. Bone, compact bone, it's very easily identifiable due to its particular preparation method, but you should be able to identify it even when it's given in HNE. So this is the central canal, and these are the layers that are surrounding it. So these layers are called lamellae, and between the two layers are these spaces where the osteocytes are present. And then this whole process, this whole lamella containing lacunae, and between them in the center there is Haversian canal containing artery and vein and lymphatics. That is all the osteon or Haversian system. So that is a Haversian system, that is a Haversian system, and you will see lamellae that are present between them. So this is interstitial lamellae that are present between them, and sometimes two Haversian canals, you may see them connected through horizontally running canals. So these are Walkman's canals. So these are the things that will help you figure out the bone, but again its feature, its color, it's very easy to identify. Spongy bone, spongy bone, I had picture open right here too. Spongy bone are trabeculae of bones with spaces between them that are usually occupied by bone marrow. So basically this so basically this is the spicule of the bone. This is the bone. The area between them is the bone marrow. So this get that, or not all of it is bone. So this is bone, this is bone, this is bone, and in between them there is bone marrow. So that is what you are seeing right here. So this is bone, this one, this is bone. This whole area is empty space between them, that is filled with fat cells and the bone marrow. And this is bone again. And if you are observant enough, you will be able to differentiate the lamellar arrangement. There is lamellar arrangement, the fibers running parallel to the surface, so fibers running in this direction. So this is the lamellar attachment that was present in the compact bone too, that is present. Osteons are not present, but lamellar attachment is there. Okay, moving on. Trachea. So you have studied most of the things before. Trachea will have pseudostratified epithelium on the top that we discussed in the earliest part. It will have haline cartilage throughout, and that is the epithelium, that is the mucosal part, that is the submucosal part with glands, and ducts of glands are seen right here. Some glands are shown right here. This is the haline cartilage, and that is the dense irregular connective tissue surrounding the outside. So that is the trachea as a whole, and this slide is usually used for pseudocertified epithelium to haline cartilage and trachea as a whole. Bronchus, that is that which tissue of the lung, and you will see these alveoli that are present right here, and that is the bronchi. And as the bronchus goes smaller and smaller, the cartilage, this is this is a very small cartilage. You can see only line cartilage here and here. So as the bronchus keep on getting smaller, the number of cartilage gets so smaller, and the surrounding smooth muscle is present right here, and the epithelium moves from stratified, pseudostratified to low columnar and cuboidal, and then ultimately squamous epithelium in the alveoli. So this is bronchus focus, but the slide of the lungs. Arteries. So let me have any pictures of respiratory system that need to be discussed before moving on. I think that's it. Mm, there are different pictures. These are alveoli with terminal bronchiole. Terminal bronchiole means the bronchiole that is the smallest one does not have any cartilage in it. These are the alveoli. This is basically this area is alveolar sac, and different alveoli are opening into it. Different alveoli are opening into it like this. So this whole central area is alveolar sac with alveoli opening into it. It's a technical alveolar duct, but this can be an alveolar sac like this alveoli that is opening into it. You don't need to differentiate this much, but it's good to know. These are bronchioles, terminal bronchioles. You can see the epithelium has become almost cuboidal or low columnar. So this is a cell that is present. So this is a low columnar or even cuboidal epithelial cells. And this is the alveoli with flattened cells between them. So that's lungs. Now arteries. Before going jumping into the arteries, let me show you a few tips and tricks I think I can help you with. Large size arteries like aorta, they have muscular part that is most prominent feature in them, and they have muscles, and the thickest part is the tunica media, that is the muscular component, and the fibers depend upon the staining. Like here, this is the, in this particular picture, this is the tunica intima, this hole is tunica media, and somewhere below it will be tunica adventitia, and on the top of that will be squamous cells, and there is a large number of fibers between, there is maybe equal number of muscle and elastic fibers. But as you move on to the muscles, muscular artery, this is an here the again the intermediate portion is thick, and the elastic component is restricted to the outside like this one, and inside like this one. These are internal and external elastic lamina, and adventitia part is small. And in the smallest arteries, in the veins, lumen is usually irregular, and the adventitia component is usually larger than a muscular component as opposed to the arteries. This is the muscular component, this is the adventitia component. You can see it more organized and more in size as compared to the muscular component. So going back to these slides, you can see different, compare the this is the muscular artery with muscular vein between them. You can see the difference between the intermedia and media and adventitia. The media and tunica media and adventitia, media is larger in size, and here you can see the media and adventitia is more prominent feature as compared to the media. Secondly, the lumen size. Thirdly, the relative thickness of the wall may give out. By the way, this is the peripheral nerve with muscle bundle and perineurium and so on. So that is the muscle, this is the arteries. And if we go through the different arteries quickly, these are the veins. And the veins, you will see the veins in the tunica media will have muscles fibers mixed with fiber. These are connective tissue. In arteries you won't see connective tissue. Like this is the adventitia, and this is the media, and in the media you won't see muscles interspersed with fibers. In the veins only. In the arteries it is very strictly filled with muscles. So that will give out the information too. That is the vein. And comparison between the two, that is an artery, especially large artery, and this is the large artery. And in the slides where fibers are not separately labeled, you may see them as not separate colored, you may see them as non-colored lines. So these lines, these are the elastic lamina. So even if you don't see them as darkly stained, not silver stained slides, you may see them going along like this. So these are the elastic fibers that are going between them. This is the artery, this is the elastic artery, elastic artery, and muscular artery. In the muscular artery you will see this internal elastic lamina that is prominent, even if not colored with a black. You may see them here. So this is a muscular artery and so on. Non-hairy skin, you will see steadfast common non-keratinized epithelium and this, and you will see keratinized, not any hair. And this is also thick skin. Thick skin is identified by a large number of cells, large number of thickness, and it is also identified by this part. This is the layer that is non-stained, that is stratum basale in the bottom. Then there is stratum spinosum, then stratum granulosum. This part is called lucidum, clear layer, and that is the stratum corneum. So this particular part gives away that it is present only in the thick skin. So you can give the answer of thick skin, and you can give the answer of non-hairy skin too. Hairy skin, you will find the hairy skin, and it is thin skin. You can see there is stratum basale, spinosum, this layer is granulosum, and this is corneum. There is no clear area between them. You can see where skin is intact, you will see no clear area between them, so there is no stratum lucidum. And here is the hair follicle with a sebaceous gland on the bottom, and here is the denser regular connective tissue that is forming the dermis and so on. And that is the hair follicles that are cut in the cross section, and you will see the hair, layer of hair itself, and the root surrounding them. So that is the one hair follicle that consists of the hair in the center with the root surrounding it. Again here, and here are the sebaceous glands that are present between them, and the hair is here, and these are the dense irregular connective tissue that is present making the stroma. So I think that's it. Have we covered all the slides? We have seen cell epithelium. Cell does not come independently. Epithelium, glands. Epithelium, we saw the simple and stratified epithelium, all the gland types of three types of glands: mucus, seromucus, and then we saw dense, regular connective tissue, and we saw the three types of cartilage: fibro, hyaline, and elastic. Two types of bones: compact and dead. Blood as a single tissue. Muscular, we have saw that um, we saw skeletal, smooth, and cardiac muscle. We saw spinal cord. I did not show you cerebrum and cerebellum. I will show you. Nervous tissue, we saw that peripheral nerve, peripheral ganglion, and in the peripheral ganglion, we differentiated between dorsal and sensory root. I will show you cerebrum. Muscular tissue, we saw medium-sized artery vein, large-sized artery vein. Lymphoid tissue, four types of lymphoid tissue. We saw thymus, spleen, lymph node, and palatine tonsil, and trachea and lungs. Now we are left with cerebrum. Okay. Cerebrum and cerebellum. Cerebrum is actually a little difficult slide to identify, but I'll try my best. Cerebellum. Let's go through cerebellum first. Cerebellum, you will see different layers, and in the cerebellum, these are the different layers, folia, that are called usually. And from here to here, this gray matter, and here is the white matter, and here to here again the gray matter. The gray matter consists of three layers, and if we zoom them, you will see three layers. Yes, so you have this layer, outer one. This is the molecular layer. This layer of large cells, this is called Purkinje layer, and this layer, that is called granule cells. In the molecular layer, there are majorly fibers, so you will see very less cells between them, less nuclei between them. And the middle layer is interestingly enough that it is made up of cells that are very prominent. So these are the Purkinje cells. These are the Purkinje cells that are making up the middle layer. These are the Purkinje cells. These are the Purkinje cells. So this is how you will recognize cerebellum. I think I have a more zoomed in picture of it. So this is the molecular layer. Outbound, you won't get pictures in this much focus, but these are the Purkinje cells that are highlighted. So that we mentioned earlier, this one and this one. And this is the molecular layer with all the three layers that are visible here: molecular layer, Purkinje layer, and granular. Granular layer is identified by the large number of cells that are present in cross approximation with one another. The nucleus will tell you how many cells are close together, and this again are fibers, bundle fibers. So this is white matter. Okay. And this is again the three layers in the opposite direction. And I think I have shared with you these slides. Cerebrum. Cerebrum, you see something like this in the slide. It has a large number of layers, but you don't need to actually remember them. Um, just see that it's the outermost part has the molecular layer, much like that of the cerebellum. Then the inner layer are not very easily differentiated between different layers, so I won't go into the detail, but you can focus and identify pyramidal shaped cells. So all of these are pyramidal shaped cell, like this one. So they help you identify these are pyramidal cells that are present in the different layers. In the different two layers there are more in number and two layers that are less in number, but the presence of this granular layer, sorry, molecular layer, and the pyramids or pyramidal cells, they have very typical shape. It's like somewhat like this. They have shape like this. You can see all of these have their apex pointed towards the top. So these are pyramidal cells that are present, and they can help you identify that we're talking about cerebrum. I think that's about it. I'll close my talk, and I hope it was more beneficial than tiring. Thank you, and good luck, lots of good luck for your examination.